Good morning, everyone. It's 8 a.m. We'll get started. My name's Kurt Langlotz. I'm a radiologist from Stanford, and this is our informatics session. Very excited to talk to you about some of the informatics ideas that are likely to support some of the problems that we identified yesterday and some of the solutions that we thought about as well. So I think it's a nice sequence where we think about the problem, classic software development methodology, think about some of the issues, and think about how informatics and information technology can at least help with some of the solutions that we talked about yesterday. We've got a great set of speakers. Unfortunately, Thomas Fuchs is ill and has laryngitis and is unable to be here with us today, but we're very thankful that Mike Bisich, who is here to be one of our panelists, will be presenting some of the material in Thomas's slot. So thank you very much, Michael. Our first speaker this morning is Amy Abernethy, and Amy is Chief Medical Officer, Chief Scientific Officer, and Senior Vice President for Oncology at Flatiron Health, and she will be talking to us about data aggregation and sharing to inform cancer care. Amy, it's nice to have you here. Thank you.
Thank you very much. It's delightful to be here with you today. I am a member of the forum, and what we thought we would do is start off this session, if I can plug in, really talking about data sharing. And as we think about the role of data sharing, it's really important to think about the data sets that we're going to need in order to prepare, for example, training data sets and other activities to help us have confident cancer diagnosis. There's been this huge conversation going on about the importance of data sharing and how that's going to strengthen our research. There's a historical definition, if we look at data sharing, that comes from NIH and others that talks about the practice of making our data sets available from scholarly research to other investigators, so basically secondary data use. Although the conversation now around data sharing has expanded markedly to think about the use of data that's being collected as a natural outgrowth of, for example, clinical care, the sensors we have on our bodies, et cetera, and the availability of those data sets to be aggregated in support of secondary research at scale. As we think about data sharing, though, we need to think about the underlying data sets and their characteristics and how we understand those data sets in order to confidently use them. I'm going to talk today about some examples from Flatiron Health, but I want to highlight the fact that the things that I'm talking about apply at Flatiron, but they apply at many of the data sharing activities that are going on across the cancer space, as well as more broadly across medicine. So, for example, within cancer, we have activities like AACR's Project Genie, ASCO's Cancer Link, the Orion data set, and then we have other national initiatives like the Collaboratory and PCORNet and the NIH Cancer Commons, and all of these have as a common underlying set of activities the importance to be able to have confidence in our data sets as we aggregate them and understand how we're going to use them for the future. There has also been increasing conversation about the use of these data sets in support of, for example, drug approvals. And what we see in 21st century cures is an increasing highlight on the role of real-world data and real-world evidence in support of new drug approvals, or at least the understanding of drugs once they're approved in the post-marketing setting. And so regardless of the purpose, so for cancer discovery and scientific research done by investigators who are federally funded, or for drug development, or for other activities, the importance of getting our data sets correct is critical. And I see this as a critical issue for our training data sets in both pathology and radiology as well. So I'm going to use this figure as a reference figure over the course of the next 15 minutes or so. And what we're looking at is a longitudinal timeline of a sample patient who is initially diagnosed with stage 2 lung cancer and ultimately passes away with the disease after receiving several different therapies in the metastatic setting and several progression events. And as we think about the aggregation of large data sets in support of cancer diagnosis, we need to understand that there's an interrelationship between the diagnostic event, the treatment event, and the outcome, and how these get curated in relation to each other also is particularly important. And I'll go through this. Before I come back to that figure, though, I just wanted to kind of show you what it looks like once we can interrelate those types of activities. And this is just one example of data sharing at scale, focusing again on lung cancer. What you're looking at is a data set of 43,000 patients who have their longitudinal electronic health record data curated. So this means that each month, any care that any patient within this data set receives is now added into that longitudinal data set, and also all new patients coming into the Flatiron Health system are added to the data set. And also this data set is linked to other external data sets. And what you can see is as you start aggregating data about patients who are receiving care in the lung cancer setting across the country, we can start to understand, for example, histology at scale. So it's been pretty consistent that about a quarter of patients have squamous cell. We know who's got who are smoking, so about 13% or so patients are non-smokers, and we're able to monitor what happens now at small subpopulations like non-smokers in the data. But also you're able to understand longitudinally what's happening across time. So for example, here what we're looking at is the uptake of PD-L1 biomarker testing. And in January 2016, over on the left, about 8% of lung cancer patients were getting tested. And now by December of 2017, about 58% of patients are getting PD-L1 expression testing. One of the important things about this story that we'll come back to, though, is that right now there's not a consistent way. There's not a consistent data model for PD-L1 expression testing. So if we're going to interrelate, for example, PD-L1 expression to the process of diagnosis, treatment, and outcome, we actually have to make up the data model on the fly. You can start to see across time, as you know, who's got lung cancer and what their PD-L1 expression looks like and what treatments they're receiving, that across time we've seen the rapid uptake of PD-1 and PD-L1 agents, as you'd expect, and that's what you see in yellow. So in January of 2016, about a quarter of patients were receiving PD-1 agents, and by December of 2017, about 45% of lung cancer patients. And you can start to say what happens in different settings. So this is in the second line and advanced settings. And so what we see is about half of patients are receiving PD-1 inhibitors, and that's been consistent over the last 2 years. But as we've seen these drugs start to be approved in the earlier settings, what you see is in January 2016, only about a tenth of patients were receiving PD-1 inhibitors. And by December 2017, 43% of patients were receiving PD-1 inhibitors in the first line setting. Why does this matter? Because as we start to think about data sets that have information about diagnosis, treatment, and outcome, you start to want to understand how can you interrelate diagnosis and outcome. Here what we're looking at is survival in PD-1 treatment by line of therapy. And we've been surprised to see over and over again that line of therapy does not predict survival, so it doesn't matter what line you receive a PD-1 in. But PD-1 expression, as we'd expect, does greatly predict survival. Now, why tell this story within the context of thinking about the development of data sets for better, more confident cancer diagnosis? All of this work first hinges on being able to confidently diagnose who that patient is and deal with the ambiguity in the underlying data. So, for example, being able to confidently understand who's got squamous cell and that underlying pathology, and figure out what to do when, for example, the pathology and its interpretation seems to change across time. To figure out how we deal with the fact that the PD-L1 expression assay has been something that is not stabilized yet, and depending on which lab you get your assay done by, the proportion of patients who are considered to be a positive test as interpreted by the physician seems to be changing across time. And so any particular PD-L1 test is a single moment in time of what the information was presented to the physician and how the physician acted on it, and that actually may also change across time. So I go back to this figure. So diagnostic events are a combination of clinical, pathological, radiological, and biomarker data that's put into context. As we think about our aggregation of data sets, we need to think about how we understand data points and context so that we can now confidently use those aggregated data sets not only to understand, for example, treatment and outcomes, but also as training data sets, for example, for pathology and radiology interpretation using artificial intelligence and other approaches. So a couple of examples. Here we've got the patient who was diagnosed in the beginning. We've got pathology, radiology, interpretation. We've got the clinical interpretation. We may also have physical exam findings. That information ultimately gets interrelated to a diagnosis of a patient with, for example, squamous cell stage 2 lung cancer. That diagnosis is now essentially a benchmark against which we're going to work from that point in time. When the patient is diagnosed with metastatic disease, we have perhaps radiologic imaging, but do we have an associated pathology report? We may or may not. Depends on what that physician did for that patient at that moment in time. How we then ultimately understand with confidence whether or not this was metastatic disease and whether or not it was metastatic disease of the same underlying primary is something that we actually interpret from what's going on within, for example, the clinical chart. This patient progresses. It progresses after receiving first-line therapy, and at that time now gets biomarker testing. And what's interesting about this progression event is that if you look over there on the right, originally the patient's progression event was ultimately called progression by the radiologists, but actually tumor response by the clinician. We had 3 different radiological pieces of information, one of which said progression, clinical notes that said, no, this is response, and another document about 3 weeks later that suggested now this patient had progression. So in fact, what was going on was we had multiple documents going on in the chart with information coming in from multiple places, needing now to disambiguate whether or not this was indeed a progression event and what was the diagnosis of going on at that moment in time. So some of the data management challenges as we think about developing these data sets is that we have to deal with the fact that what seems like specific and clear events in time, a diagnosis, a metastatic event, a progression event, for example, actually can be quite ambiguous. And that ambiguous information has to be dealt with in the context of our shared data sets. We often talk about the need for interoperability, but a lot of the need for interoperability is the need for an interoperable common data model, or at least translation steps that help us work between data models. Our data sets need to be curated in order to be able to be cleaned up and dealt with at scale. This ultimately is a big problem as we think about electronic health record data that have a lot of unstructured documents. We need to be able to identify the source documents and be able to trace back to source when we need to go back and understand exactly what a piece of information really was. There's differences in timing of assessments, which is an interesting problem in data sets, which I won't get into much today. And then we need to be able to think about provenance, traceability back to source, and all the standards that go behind these data sets. I'm going to give you a couple of examples of how we're dealing with that. But ultimately, my core point here is that the way the data sets are aggregated and curated is very important as we think about having data sets that are ready to do many of the important examples that you're going to see in a few minutes. So at Flatiron, what we do is aggregate electronic health record data. We process both the structured and unstructured data coming into a common data set and then link that with other data sets. The curation of the information then has to be done in a very reliable way. And at Flatiron, we've spent a lot of time focusing on how do we curate that information, how do we pull together what's in the pathology reports, what may be ultimately inaccessible tissue, what's in the radiology reports, what may be ultimately inaccessible images, and what the clinician says and what's going on to actually now come up with what the initial diagnosis was and how we're going to understand with confidence what that initial diagnosis looks like. At Flatiron, we do this predominantly using human curation, in other words, real people who pull out this information out of documents. Some groups are working with artificial intelligence and other means. This is just an example of how we curate data out of, for example, a PD-L1 IHC report into a single common data model. And that common data model becomes an important issue as we think about aggregating data sets across time. What's interesting is that probably the most easy part of this data model is the date that the tissue was assessed and the final result positive. Outside of that, the model is not really common to any other data models out there because one single model does not exist. We use a process we call technology-enabled abstraction. Ultimately, my core point here isn't how we do it at Flatiron, but that there actually has to be a systematic process in the background that you can understand about how data sets are curated. Because if you want to have confidence in the data sets that you're working with, you need to be able to look under the hood and understand how you got there. And then you need to be able to document back to source and maintain full provenance. What does that mean? So, for example, in this particular progression event, that progression event was abstracted out of the chart of Jane Doe, and it was abstracted by a single person who looked at source documents from a single electronic health record down in Florida. And as that person was abstracting that information, we need to know, what did she look at to abstract the information? How was she trained? How good is she compared to her peers? We need to understand, when she pulls that information out, how confident are we that that particular piece of information does represent a progression event, and how confident can we be across time? And that ability to maintain consistent back-end information and get all the way back to source documents is a critical part of well-curated data sets. We do that by a system of quality controls at Flatiron that really look like clinical trials quality controls. Either way, the system to which you're doing your data curation and you're maintaining your data sets become particularly important, especially as these are training data sets for artificial intelligence. And this is just how we document completeness and data quality. And data linkage then becomes possible because you can, for example, link to mortality data, claims data, patient-reported outcomes, et cetera, all coming together all around a particular data set and patient. So then what this does is give you a comprehensive view as you pull data sets together of individual patients. And this now allows you to have a confident understanding of what happened at a diagnostic event, what were the treatments related, and then what happens to that patient, which provides that interrelationship across time. We've been doing this in collaboration with the FDA, and part of the purpose of this collaboration is to ultimately be able to document approaches to describing quality of underlying data sets, because if we can describe the quality of the underlying data sets in a consistent and confident way, we'll be able to do that across different kinds of shared data sets and different kinds of data sharing activities. And so that's been a key purpose of our Flatiron FDA collaboration. We've also been describing this in the literature, and I just want to point to this book of work because, importantly, one of the things that we need as entire sort of data sharing community is a consistent terminology to how we think about data quality and the characteristics of our data sets. And that's one of the things that we've been working on publishing. We call this the real world evidence or real world data quality checklist, which basically gets to some of the things that we need to describe in these shared data sets. So we need to be able to describe data completeness, data reliability, how long is longitudinal follow-up, what was the quality and the underlying controls, things like timeliness and recency. We also need to be able to describe issues like generalizability and what the full provenance for the data sets were. In the last couple minutes, I just wanted to hit on a couple data quality concerns that I'd like you to have in the back of your mind as you listen to the next set of conversations, many of which we've already hit upon. First, there are challenges with images. First of all, raw images are not always available for review, and deciding what you're going to do in your data sharing consortiums mean that you ultimately need to figure out how you're going to deal with times when you're looking at reports versus having available images. Certainly one of the things we all know is that not every scanner is the same. And so differences in scanners, differences in images mean that we run into problems with differences in being able to assess key endpoints, for example, like RECIST. There's changing interpretations over time. It's not just in changing pathology interpretations over time. In our lymphoma data sets, what we see is about 15 to 20% of patients have changes in their lymphoma assessment and interpretation over time, but we see it also frequently in our radiologic interpretations. There is no consistent approach out there for documenting data quality for shared data sets like this, and this is one of the areas that we think is a critical pre-competitive set of activities that needs focus. And there's no consistent approach to how we document endpoints in these data sets beyond what we think in traditional clinical trials, such as RESIST, and that's another thing that we've been focusing on. We need gold standards. So for example, at Flatiron, we use the National Death Index as our mortality gold standard. We don't have national gold standards for many of the data points in these data sets, and that's something that we hope that the National Academy can help to shepherd forward. And we need to also think about how data in context helps to reinforce diagnostic accuracy. If we can understand, for example, whether or not a PD-L1 expression assay predicts response to PD-1 and PD-L1 agents, that helps us understand better both the diagnostic accuracy of a PD-L1 expression test, but also how different tests perform against each other. That then takes us to the availability of data sets to do many of the really awesome things we're going to hear about in just a couple of minutes, which are confident data sets that allow us to have labeled data for artificial intelligence and machine learning, and basically to train our algorithms as we start to think about many of the things we can do in the future. So with that, the problems that we're solving, ambiguous interpretation, differing issues between timing and scans and how that influences, issues around data curation standards and appropriate labeling, problems around data linkage, you notice I didn't even get into privacy today, but that's a huge issue. A consistent approach to documenting data quality, consistent endpoints, and appropriate use of AI and machine learning in this overall space. So with that, I'll say thank you very much, and I look forward to many interesting conversations.